All right, so we have Steve from MSc Molecular Medicine, and his interest is in bacterial genomics, immunoinformatics, and drug discovery. All right, that's pretty interesting. Can you also let us know from which university you're from? And what about the rest of you who have joined us? Can you also introduce yourself in the chat box? Yes, hello Russell, it's good to have you back. So Russell is a research assistant in Genome Center at Joshua, and his research interest is in metagenomics. Thank you so much. I'm curious to know, can you also let us know what is your motivation to join the field of research? Is it as a part of your degree, or do you plan to go for PhD or? All right, Steve is from Jomo Kenyatta University of Science and Technology and is currently working as a bioinformatician. All right, perfect. Thank you so much. And how would you rate your skills in bioinformatics? This is a general question for all the participants. Are you a beginner or intermediate or advanced expert? All right, so Russell says he has intermediate knowledge. Thank you so much. And Steve says there's a junior bioinformatician and intermediate skill. All right. So we do have a couple of informatic experts amongst us. And what about the rest of you? I'm curious to know about you guys as well. Tanmoy is also intermediate. Tanmoy, can you also introduce yourself in the chat box? Like the rest of them, you can let us know about your educational background and research interest. All right, I can see that few more participants have joined. And for those of you who have just joined us, we'll be starting with the webinar in just 2 to 3 minutes. And meanwhile, we are waiting. It would be great if the participants can introduce themselves. Few of the participants have already started introducing and letting us know about their research interest. All right, so we have response in chat from Tanmoy. He is a current fourth-year student of microbiology and is interested in DNA-specific variants. All right, thank you so much. And what about the rest of you? I think almost everyone has covered. All right, thank you so much for your patience. We will be just starting in just a few more minutes. Thank you so much. All right, so without further ado, let's start with today's webinar. Hello, everyone. A very warm greeting to one and all. My name is Srigauri Krishnakumar, and I would like to welcome you all for the webinar on OmicsLogic, Bioinformatics and Data Science Research Fellowship. The Research Fellowship program has been designed to help young researchers and students take advantage of the bioinformatic resources for the analysis of complex life science data and become well-versed in bioinformatics. Research fellows participate in cutting-edge bioinformatics research led by expert mentors, and the fellowship program will offer a combination of online resources and mentor guidance to prepare you and help you complete a bioinformatics project. So before we begin with today's webinar, for those of you who are joining us for the very first time, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of our mission, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by over 24,000 participants from 187 countries in over 300 workshops in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, and data science for biomedical data. And as a part of the training, you will also have access to the OmicsLogic resources, which takes a combination of training modules, data analysis tools, curated project data sets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. And this also comes with an access to the OmicsLogic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. The OmicsLogic platform also give options for analyzing a given data set. And these data sets and associated publications can be used in several ways. For instance, you can learn bioinformatics without any coding at all. And for those interested in opting the non-code pathway, they will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. And the platform includes demo pipelines, as well as data management and analysis cloud infrastructure to run the bioinformatic pipelines. And different stages of analysis are performed in different sections on this multi-omics platform. And for those interested in learning to analyze data through coding, we provide the OmicsLogic Code Playground, which will help you develop coding skills while learning popular packages in R programming and Python to visualize, annotate, and analyze complex patterns in biological data. So now, to view the various courses, example projects, and student projects, you need to first sign up on the OmicsLogic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles, a brief bio about yourself that will consist of your educational background and research interests. The mentors can view your progress under the Activity tab. The courses you are currently completing will appear under the Courses tab. Finally, after the completion of your coursework, the certificates will appear under the Certificates tab. So that was all about how you can update your profile. So with that, let us begin by signing up on the OmicsLogic Learn portal. With that, let me paste the link for signing up in the chat box. For those of you who have not signed up on the portal yet, please visit the link and I'll walk you through how you can sign up. All right. Russell has shared a query. I'm sure one of our team members will look into your issue and get back to you. All right. So that was the link for signing up. So for those of you who are signing up for the very first time, head on over to the link and click on the Create an Account option. So once you click here, you can sign up either by entering your name, providing email ID and a password, or you can also sign up using any of your social media handles. That could be your Google account, Facebook account, Apple account, GitHub account, or even Twitter. So that is how you can sign up. And for those of you who already have an existing account on the portal, you can simply click on the Login Now option that you see here, and then enter your existing email ID and password and directly sign in. So I'm monitoring the chat box for your responses. Once you have signed up, please put it in the chat box as a yes, so that I know I can proceed ahead. I'll pause for a couple of seconds, and after that, I'll head on over to show you how you can navigate through the portal. Thank you so much, Nurani, for confirming. And if you're facing any technical issues, you can also let us know. And our team is here and we'll be happy to look into it. All right. Thank you so much, Tanmori. All right. Without further delay, I'm just going to sign in. But along the way, if you need me to repeat any steps, you can let me know and I'll be happy to show you. And since I already have an account on the portal, I'm going to click on sign in. And once you sign in, you will be able to see the courses page. And when you scroll down below, you will see a search bar that says looking for a specific course. So here, if you enter a keyword that is of your research interest, that will show you the various courses and example projects that make use of that concept. So for instance, if you are interested in learning to analyze transcriptomics data sets and you want to get started, you can enter the keyword transcriptomics, and that will show you several courses on bulk RNA-seq, single-cell RNA-seq, and even analyzing transcriptomic data sets using R and Python programming, and several other example projects that makes use of transcriptomic data. And now coming to the part about setting up your project, you can click on the blue bar that says welcome back with your email ID. When you click here, it will directly take you to your account settings. So here you will see a button that says profile. When you click on this, that will enable you to edit all the features that I was talking about in my presentation before. So just as a reminder, make sure that your name is in proper capitalization. You add a profile image for yourself, add a bio consisting of your education background and research interest, as well as update your social media handles. In my case, I've linked my LinkedIn account and my Org ID. And then you will also see several other tabs in your account settings that consist of activity tab, wherein community manager and the program mentor will be tracking learning progress. You can take the course progress review under the courses tab. Under the projects tab, it will display the research project once you publish. And then another interesting area that I want to show you is the projects tab, wherein you can take a look at all of the student projects that have been carried out on a diverse range of fields, from infectious disease to neuroscience to machine learning to oncology. And so I'll paste the link in the chat box for the participants once so that you can navigate through and take a look at it at your pace. So that was all about navigating through the OmicsLogic Learn portal. So now to learn more about the research fellowship program and the various specialization tracks, I'd like to pass on the stage to one of my colleagues, Sonali Kal. Over to you, Sonali.
Thank you, Shri Gauri. Screen. All right. So before I really start discussing with you all about the different research specialization tracks that a research fellow can work on with us, I really wanted to know about the backgrounds from which you all are. By background, I mean, are you from biotechnology field, a bioinformatics field? And some of you might have already figured out which specific field they want to carry out their research on. Some of you might also be learning about these different fields and then would figure out which field they want to pick. That's perfectly fine too. Let us first of all see how many of you are from biotech field or bioinformatics or biochemistry. Can you please write down your research backgrounds or educational backgrounds in the chat box, please, so that I know we have our audience from which all fields. Let's keep this session more interactive. So we have kept this session for you all today so that we can learn about your interests and we can answer any questions if you have in the direction related to your interests and the field that you want to pick. Noorani is saying microbiology. Shapon is from pharmacy field. Okay. Saqib Hussain is from biochemistry and molecular biology. Calvin, Darius, Gyodate, Hidayah, Janatul. We have Alpana and Akanksha also with us. I know your fields. Madhvi has also joined. Hi, Madhvi. Hi, Rob, Parthiv, Pratiksha, Professor Anovar, Rasid, Saqib, Shan, Tanmay. We have people from bioengineering and chemistry field as well. That is interesting. Have you thought of the research field that you really want to work on? So for people from pharmacy, I can assume they are interested in cheminformatics or structural biology. Or if it's another field, you can tell me that as well. I just want to understand what specific research track do you want to work on. So maybe you can answer my question in a way related to the specialization track. So when we talk about omics data analysis, we basically refer to different types of omics data techniques. Like we have transcriptomics with us, or genomics, or metagenomics, or epigenomics, or proteomics. Right? So bioinformatics basically deals with these different omics data analysis. Now, when you want to work with these different omics data analysis, there could be different research specialization tracks or fields where you can apply the knowledge that you gain from these different data analysis techniques. The basis obviously remains the same, where for genomics, you're interested to look at specific variants, mutations, or understand phylogenetic trends or evolutionary trends, look for pathogenesis. And then for transcriptomics, you are more interested to look at specific types of genes, annotate their pathways, then maybe repurpose drugs which could effectively reverse the differential gene expression of specific set of genes. Metagenomics, you would like to look into the abundance levels and classify taxonomy for different microbial communities. Epigenomics, you want to understand the environmental factors responsible for a specific type of condition in hand. So the different types of data analysis we understand, but what are the different types of research tracks where these different analysis types could be explored? One is infectious diseases like Ebola, malaria, SARS-CoV-2. And I am right now on a research project topic page, actually. Here is the link. You can take your time and go through it. I'll just discuss this program page with all of you, and then I would love to know what are the fields that you want to work in, so that we can really discuss how we can proceed one step ahead and start with a research proposal. Okay, so the first thing that I mentioned is infectious diseases. Now, on this project topics page, what we have tried to do is we have tried to put together these different research specialization tracks, and then the types of data analysis that is possible for you to work on. And we have just listed some questions that you can answer maybe for these different types of data analysis techniques. Obviously, there are a lot of possibilities. We have several questions as well. And if you are a student, a BTech, BSc, MTech, MSc student, or a PhD scholar or a researcher from a wet lab domain or field, and you want to get your hands dirty with bioinformatics analysis, and you're wondering what type of research topic would you like to work on, you can explore this page and come up with the research interest that you want to really work on, and then we can get started. That is the main objective, to have this page put together. Now, a lot of students come to us for working on their dissertation projects, and they have one type of question. So we have two types of groups. One group, they know what type of question they want to work on because they have a specific type of research interest already defined. Another type of group, they have an interest, but they're not very sure how can they start working on the research question. Or there are also students who are from wet lab fields like biotech, microbiome, biochemistry, or medical sciences, and they want to learn about bioinformatics, and then they want to understand the scope and the fields, and then start working on a specific type of research question. All right, so for these different types of people, we would first of all take you through an in-depth knowledge or an understanding of these different data analysis. We'll then pick a specific type of data analysis and give you a specialized training on it, and then pick a research field that we want to work on, applying the data analysis technique skill set that you gain from your training. Now, the different research specialization tracks that are available for you to work on. One is infectious diseases. On this project topics page, we have put together the example projects as well that you can go through. Now, what are these example projects? If you go to the OmicsLogic Learn portal and click on the courses tab, you have several courses here. That is one thing that you can go through and upscale your skill set. And apart from this, we also have some case studies or projects. These case studies or projects will help you understand how to answer a research question, a specific type of research question, using a specific type of skill set. So if it's transcriptomics, you want to see what type of projects do we have here with transcriptomic data analysis. So you will get the list of these projects up here. You can open up any one of them And these are short case studies for you to get a good taste of how research questions could be addressed. For beginners who are new to this field and would like to learn, they can go through these case studies. Now, on the project topics page, what we have done is we have put together these different example projects or case studies, I will say, that we have available on the portal. They are hyperlinked with the links of their specific case studies. You can click on them and go through them to have an understanding. The second type of research specialization track that we are working on extensively is oncology. And for the field oncology, there are different data analysis types that you can work with, be it transcriptomic, genomic, metagenomic, epigenomic, structural biology, and cheminformatics. Also, there is a possibility to do a multi-omics integration and metagenomic data analysis under the field of oncology. Now, there are several different research question types that one can answer, and all of them are listed out here. You can take your time and explore this project topics page at your own pace. Some example projects related to the field, which are on cancer macrophages, PDX tumor microenvironment, and we have a TCGA study where the data was taken from TCGA to understand or predict the liver cancer risk. The next field that we have is precision medicine. And for precision medicine, again, different types of data analysis techniques that are possible, be it transcriptomics, clinical data analysis, epigenomic, and genomic. And an example project related to precision medicine. And then you have the field agriculture, very interesting field that is also gaining a lot of interest by bioinformaticians. And also a lot of agriculturalists are interested to now learn how bioinformatics could be used for working with the data from plants and crops, and also understand the association between microbiome and the plants. So the analysis possible becomes genomic, transcriptomics, and metagenomics. So that was about the field agriculture. And then you have neurosciences, where you can apply transcriptomic studies. Also genomic studies and microbiome and metagenomic studies are possible as well in depth that you can work upon for Alzheimer's. And then you have metabolic disorders, where again, the transcriptomic studies could be explored, and space omics, where you have the fields transcriptomics, epigenomics, and metagenomic data analysis that could be done. Now tell me in the chat box, everyone, what are your areas of interest? If you have figured out, even if you have not, you can let us know in the chat so that we can discuss with you a one-on-one about your interests and get you started. So let us begin with Akanksha. Akanksha, what is your research interest? You can put in the chat. Aryansh, I think it's you, Alpana. Please put your interest as well. Clement, Darius, we would like to know what are your areas of research interest. You can put them in the chat. I'm waiting for responses. Hidayah is seeing neuroscience, epigenomics, perfect. Janatul, Rob. Thank you, Hidayah and Darius, for your reply in the chat. Infectious diseases, but you want to go through. Perfect. Clement, yes, I remember. And Akanksha is interested in multi-omics data analysis and NGS molecular aspects. Okay. Now, some of you might also be interested to learn about the different possibilities of research questions that could be addressed using these different types of data analysis for the fields that I just mentioned about. For that, you can also explore some of the existing research projects by our research fellows. They have completed these projects and they've uploaded them on the OmicsLogic Learn portal. You can click on this projects tab right here, and you'll be then redirected to these different projects by our research fellows. So you'll get to know how they went through a specific type of data analysis technique and then picked up a research question and addressed the same. So you can pick up the specific type of field that you are interested to explore, transcriptomic data studies for oncology, and you'll get these projects listed out. You can open them. And just like a publication, they have all the different sections of their project added here, the data set that they explored, abstract, introduction is given, methodology, results, discussion, and conclusion. Also, one of the questions that we hear the most is, what about the data? What specific type of data can they work with? So there are two types of individuals, I say. One cohort or one group is such where they are generating data in their own lab and they're interested to analyze it. And the other cohort could be the one who are interested to learn a skill set and apply it in a specific type of research field, but they don't really have the data. So for them, you need not worry. There are various public repositories from where you can explore data and extract some specific sets that you would like to work on, specific samples, and then prepare your data, process it, and then start analyzing it. So we leverage a lot of public repositories. We take the data from there based on the type of a research question, interest you have, and then start working on it. Apart from the research topics that you are sharing in the chat, please feel free to unmute yourself, and you can also ask any question if you have related to a topic or a field, how you can work on it. And for those who are from different wet lab fields, as many of you mentioned, you're from biochemistry field or microbiome, and also we have participants from pharmacy field. For those who are new to these different omics studies altogether, you can go through the introductory courses. The first course is Introduction to Bioinformatics, which will give you an understanding of what the field bioinformatics is, what is the role of big data in bioinformatics, and also the role of bioinformatics in healthcare. And it's a very short coursework. It will give you a good, nice understanding and introduction to the field bioinformatics. And after this course, you can go through Bytes and Molecules. Now, this course, Bytes and Molecules, will help you understand or get introduced to different omics fields that I just mentioned about. So if you are new to the field bioinformatics, you can take up these two courses. The first one will lay off a good base or a background for the field bioinformatics, and the second one will give you a good understanding about different omics fields. It's definitely not possible and also not important that you take up all the omics fields all together to get a specialization into. You can pick one specific omics field that you are interested to get an expertise on and start with its training, and then work on a research project on it, and then maybe pick up the other field. So to understand what type of data analysis you want to work with and what type of research specialization track you want to work on, you can go through this course on Bytes and Molecules. So all of these different research specialization tracks that I took you through, we have them introduced in the Bytes and the Molecules coursework as well. So if you are new to the field precision medicine, but you have heard a lot about it and you want to understand what are the different scopes of research in the field precision medicine, then you can take up the lesson 11 in course 2 Bytes and Molecules to understand the scope and application of precision medicine, and so are the other fields of for research that are there in the course Bytes and Molecules. All right. So that is all that I wanted to mention. I just wanted to introduce you to different omics data fields and different research specialization tracks that you can really work on with, and how you can identify the area of research interest that you want to go through. Take some time. You have the link of the research project topics with you. You can explore this project topics page, and we'll share our email address with you as well. If there is anything that you want to discuss with us, for any guidance, you can reach out to us and we'll be there to assist you with your questions. And with that, I would now like to pass on the stage to Sri Gauri to invite our presenter for today, Ms. Harshika. Sri Gauri, over to you.
Thank you so much, Sonalika. So before we move ahead, let me quickly share my screen and show you some of the various outcomes that we have as a part of students completing their project. So as Sonalika mentioned, we do have several projects that are published on the portal. But apart from that, there are several other possibilities and outcomes that the students have showcased after completing the research fellowship program. So to begin with, majority of the students are focused on publishing their work. For instance, Rupi Vaja, a biomedical science graduate student, she worked on assessing the underlying reason for development of gray matter lesions in multiple sclerotic patients. Further, she went on to publish her work in Immunogenetics, which is an open access journal that focuses on genetic research areas of autoimmune disorders. And another interesting student publication is that of Supanta De, a genetic engineering student. He studied the effect of malaria vaccine on the transcription profiles of patients to determine its efficacy and understand the molecular mechanisms underlying the protection that the vaccine offers. He also went on to publish his work in Genomics and Informatics, which is the official journal of the Korea Genome Organization. Presently, we also have several other students who are working on their publications, like Nivedita Arambam, who is working on transcriptomics study of cancer treatment on patients and its after effects leading to relapse. Like her, we are looking forward to several other publications from our current students. Further, we also have the opportunity to participate in poster presentation competitions as a part of the Omics Research Symposium to win exciting cash prizes. The student projects are not just limited to graduates or doctorates. For instance, Andre Muadano, a high school student at Mexico, presented his neuroscience project at the Regeneron International Science and Engineering Fair, for which he won the Potamkin Prize and the 4th award in the biomedical and health sciences category. And this program can also be integrated as a part of the existing university curriculum. For instance, Pine Biotech offered machine learning for biomedical data, course which was integrated into the systems medicine graduate program at Georgetown University. And the lectures were prepared considering the interest of the program director. And the training ended with students submitting their research projects on the OmicsLogic Learn portal. And these project topics range from infectious diseases, metabolic disorders, neuroscience, psychiatric disorder, oncology, machine learning. All of these projects are up on the portal, and all of you can have a take a look at it. And these projects can also be showcased for higher studies and international university application. Like some of our research fellows, Dilara Daikin, she got accepted to the Erasmus IMBRC program, and she is presently majoring in marine biology. Dhruv Mehra, he is a precision medicine graduate program student in Manchester University, and Anusha TA got accepted to the PhD program in metagenomics at the University of Sweden. Faculties and researchers have also collaborated through our programs to publish their research work, and our latest collaboration has been with a team of researchers at Noguchi Memorial Institute for Medical Research at University of Ghana to carry out a computational-based study to understand variants associated with sulfadoxine resistance in Plasmodium falciparum. And it is also possible to pursue an entrepreneurship and business innovation track, like a team of industry researchers from Biologic Support, from a biotechnology company in Nigeria, And their aim was to improve the fertility of women in Nigeria and improve their chances of carrying their babies to term by developing a kit test. And the entrepreneurship track is not only pursued by industry researchers or academic researchers, they're also pursued by students. For instance, a group of undergraduate and postgraduate students worked on developing an early detection kit as a part of their aim to identify and detect neurological disorders with a specific focus on Alzheimer's disease. So all of these are just very few examples that I just wanted to showcase among several other outcomes that we have seen. And with that, I would like to pass on the stage to Harshika to hear from her about how she has carried out her research project on rheumatoid arthritis. Over to you.
Hello everyone, I hope my voice is clear and audible. So I'll share my screen.
Yes.
Hello everyone, my name is Harshika and I was a research fellow student at Pine Biotech. My project was mentored by Dr. Raghav sir. My topic of the project was comparative analysis of RNA expression and M6A methylation in rheumatoid arthritis. So before starting the projects, what these are the courses that I've been through to finalize and come the to select a topic. So first of all, I just were casually understood about the introduction to bioinformatics. So this course gave me a brief outline about different sectors that are present in bioinformatics, where and all bioinformatics will be used or is currently used. Everything was given in this course. Then, uh, then coming to the specifically data in life sciences. What is data and how it is, how they produce data from sequencing, different type of sequencing, and why it is important to store data, why it is important to analyze and study those data, not just single data, multiple data are taken. So why it is done and its advantages, its applications, everything was given in this course. So after that, I went to the next course wherein designing and bioinformatics project. So in this project, a clear brief view was given how to select a topic, how to do a literature review, and what are the things you can look into while selecting the project. And when you do the literature review, you'll know what is the recent work which is done in a particular disease. For example, I selected arthritis. So what type of data they are taking, what type of analysis they are doing, and which a part of or which area of the analysis is not discovered or understood more. So we'll know about all these things when we do literature review. So after that, doing the literature review, so I wanted to analyze the methylation pattern and also the normal RNA expression pattern. So I just quickly went through this epigenomic course before starting with the transcriptomics because as we all know, epigenomics was study of mechanism that causes changes in the gene expression, but there is no change in the DNA sequences. So this epigenomics plays a major role in studying various diseases. Then in the transcriptomic course, starting from how we should select the data, how the data is pre-processed. So usually when a data comes from sequencing, in the lab when they send for sequencing, adapters are attached. There are a few duplicates when we sequence. There may be few duplicates. So to remove that, what are the pre-processing steps are done about that we studied. Then how to map So we have a set of sequence. Then we choose to map it with the sequence which are already present. That is mapping with the genome or mapping with the transcripts. Sorry. So these things were explained. Once we get the data, so after we select few samples, for example, in my study, I took 15 samples. So in that they check whether all the sample are showing variation. Only when it shows some amount of variation, there is a use of analyzing it. So one mean of testing that is principal component analysis. So this was the brief of what is done in transcriptomics, that is analyzing the gene expression. So after there is a one tool called DSEC and EdgeR. So this tool basically gives you the differentially expressed gene. Means in a disease, some of the genes are upregulated, they are expressed high, or some of are downregulated, they are not expressed where they should be expressed. So this to know this differentially expression analysis is done. Then at last, this BioML gives you a detail about how to do statistical analysis starting from the t-test. And when in the machine learning process, which I used in this project was from the T bioinfo server. So what happens in the each step and how to interpret those data. We may get the data very quickly within few days, but it is also important to interpret the data. So how to interpret it, how to know whether this data is giving some amount of good information and everything. So yeah, this was all about the courses. Then before going to the results, I would like to give some introduction about rheumatoid arthritis. So it is a chronic inflammatory joint disease which causes bone damage and cartilage damage, which when left untreated ultimately leads to disability. Primarily, the synovial joints are affected because the cells attacked by pathogen and our own cell, our immune cell fails to differentiate between this and they attack our own cells. So yeah, it is a type of autoimmune disease. So usually the emergence of RA or rheumatoid arthritis is apparent after the immune cell activation and proliferation, including T and B cells, monocytes, monocytes. So these cells, when they see that there are high amount of proliferation or when they are circulated in high amount, they confirm that there is a RA, specifically in the synovial membrane. So for now, there are different drugs used for managing RA, which includes disease-modifying anti-pneumotic drugs. However, a significant amount of patients do not respond to these drugs. Because the combination of RA, its occurrence is not only genetic, it's also environmental and mainly the epigenetic factors, uh, which causes the variation in drug response. So for my study, I took modification called M6A, which is an epigenetic modification. So I also mentioned earlier what is epigenetics. So it is a study of heritable and reversible changes in gene function, which do not involve any changes in DNA sequences. And the three important epigenetic modifications that are widely studied is DNA methylation, histone modification, and non-coding RNAs. So in my study, I took M6A methylation which was altered in arthritis. N6A is nothing but at the sixth position of the methyl in the N group, the methyl group is added. So it is the most abundant post-transcription RNA modification. This modification occurs both in coding and non-coding RNA. So yeah, the it involves methylation at the sixth position of only the adenine bases in the RNA molecule. The aim and objectives for my project was to study the differentially expressed gene and dysregulated methylated peaks in pneumatoid arthritis. So I studied both of the data. Um, there were 5 data which were used for studying the differentially expressed gene, and the other 5 data with the control was used to study the M6A methylated peaks. While doing this, our aim was to find the common genes that were enriched. Enriched means which was differentially expressed in RNA-seq and MERIP-seq, which is nothing but methylated RNA. Immunoprecipitation sequencing is the method by which this data was generated. And after finding the differentially expressed genes in both of these groups, which pathways were enriched were found. And then to understand the importance of RNA methylation, a post-transcriptional modification, and how it causes prognosis in RA disease. So here is a brief outline of my project. So I selected this bio project under the code PRJNE795351, which is available in NCBI. Then I explored the data to find variations and if there were any outliers using principal component analysis. Then I used these tools, Trimomatic, PCR Clean, HitSat2, HeadSet-seq tools, which was run on T Bioinfo server. Then after that, I studied differentially expressed gene using a dissect2 tool for the input and IP sample. So after pre-processing all my data, I did separate study on input, which is the RNA-seq data, and IP here refers to the M6A methylated data. So now I got 2 groups. After studying it, I could find some of the gene which were upregulated or in the case of methylation they mention it as hypermethylated. So some of the genes were present in both the groups, like they were common, and also similarly downregulated and hypomethylated. So as I could see that there was some amount of gene that were both common in this, I did an conjoint analysis to find to separate the all of these common genes again into 2 groups, wherein upregulated and hypermethylated one group, and other group was downregulated and hypomethylated. I would like to mention that hypermethylation means when more amount of methyl group is added or present in the adenine base. And hypomethylated is when the methylated groups or methyl group is removed from the adenine base. So for all of those genes which were found to be common, I did gene ontology and kept enriched pathway analysis to find the significant pathways that were associated in causing RA disease. So here is the graph of the conjoint analysis I did, and the top group, which on the right side in orange color refers to the gene, only the top genes I took. So out of which after up to the 1 and 1, I could find 185 genes that were in this group, which is hyper and upregulated, and some with the names mentioned here are the top genes. I've also written on the right side, and similarly on the downside you can see 139 genes that were showing hypo and down regulations, and these were the top genes. So I took those genes and I did a gene ontology and kept pathway analysis. So what are the main pathways these genes which showed high enrichment here? What are these genes, which pathways they are involved in? So when studying that, I could find the top pathways, and I differentiated them into this tabular column as you can see here. Here this is for upregulated and this is downregulated and hypomethylated. So upregulated genes showing enrichment involved in these pathways. This is not, I would not say this is exact, but yeah, somewhat. So they were intracellular receptor signaling pathway, somatogenesis, negative regulation of a type of apoptotic signaling pathway, then positive regulation of cytokine and neurogenesis, then response to oxygen radical, positive regulation of intracellular signal transduction, and leukocyte apoptotic pathways. So all of these pathways, genes involved in these pathways were upregulated and hypermethylated. And genes involved in these were downregulated or they were not functioning. Basically, that what it means. So the pathways were cellular response to leukemia inhibitory factor, actin filament polymerization, positive regulation of GTPase activity, acetyltransferase activity, sensory perception of mechanical stimulus, recycling endozyme membrane, and epipherin binding. So, with this, I could conclude about the RA disease that it's the most common autoimmune disease and it is characterized by immune cell infiltration, fibroblast like synovial cell hyperproliferation, and cartilage and bone destruction. So, to date, numerous studies have demonstrated that immune cells are one of the key targets for treating RA. But looking at the M6A methylation, which occurs as an internal modification in the eukaryotic mRNA, this type of modification is not only involved at the starting stage. This M6A or methyl groups are added, removed, and all the process involved in by starting from splicing, its stability, export, and also degrading of RNA metabolism. So it shows that this methylation, specifically M6A, is changed in all of these stages of RNA metabolism. They play a significant role in various biological processes such as viral replication, cell differentiation, by controlling gene expression. So, the relation between M6A enzymes, immune cells, and RA suggests that M6A modification offers evidence for pathogenesis of RA, which will help in the development of new therapies for RA. Therapies which are specifically targeting this M6A can give us more brief treatments, because as you know that methylation pattern or epigenetic regulation is reversible. As these methyl groups are added, they are also demethylated or the methyl group is removed. So as they are reversible, they can be targeted for developing new therapies. And RNA sequencing showed that multiple microRNAs contained M6A modification. Therefore, M6A modification is important for the biogenesis and stability of at least some microRNAs. In my data also, so here on the right side, I could find one microRNA which was showing hypermethylation. So there were many, but in the top I could find only one. So this also shows that we can, study this microRNAs, and it is also important for, biogenesis and stability of some of the microRNAs. Extensive research on the methylation mechanism of M6A has demonstrated that regulation of RNA level related to M6A is complex and diverse. We believe that further research on the mechanism of N6A methylation of RNA and microRNA in pneumotide arthritis will have profound implication for treating immune disease, specifically arthritis. So yeah, I would like to thank Find Biotech and OmicsLogic for providing me this opportunity to present my work. Thank you.
Thank you so much, Harshika, for explaining your research project in such an extensive manner, and all the best for your future endeavors. So, now, I would like to tell you all about the research fellowship program which Harshika was a part, and what kind of journey she followed or a structure she followed so as to complete her research project. And then I'll quickly explain about the resources which one would have access to once he or she enrolled in the program, and then what are the fee structure and the scholarship available for everyone. So let me share my screen. I guess it's loading. Okay, so can anyone confirm if you can see my screen, like my screen is visible?
Yes, Parsh.
Thank you, Shri Gauri and Harshika. So this is the landing page of OmicsLogic Bioinformatics and a Data Science Research Fellowship Program. I'm pasting the link here in the chat box so that everyone can go through once the webinar, the meeting is over, at your own pace, and look through the page, what all resources, then what kind of structure we follow, what are the possible outcomes one would have when they enroll in the research fellowship program. So this bioinformatics and data science research fellowship program is not only for those who are like kind of in their intermediate or an advanced level and they have a good thorough knowledge of bioinformatics or data science. So this research fellowship program, as you can see, it's like it's been designed for all the young researchers who are even in their beginner level and they don't have any kind of knowledge related to bioinformatics or data science or any other omic field or any kind of specialization track. So in this research fellowship program, we offer a combination of online resources and mentor guidance so as to prepare you and help you complete a bioinformatics project. So once you scroll down, you can see that there are three different steps which we follow so as to complete the research fellowship program, and you can come up and make you ready with your research project for the publication part as well. So in a very layman language, I'll describe all the three stages which one has to has to go through. So the very first stage is get ready through training. So in very simple words, like we will train you, we will train you according to your research interest through our OmicsLogic Learn portal. So as the name is depicting, there are training, it's a training and a learning portal. As Sri Gauri has already showed you what are the different kinds of lessons or example research projects or case studies through which you can which you can go through and gain those conceptual as well as theoretical knowledge which will help you in the second stage, that is doing some exploratory analysis, or you can say literature review, and then preparing a full methodology which you need to follow so as to come up with some results and then do the interpretation of those results. So the very first stage is to train you, whether you are just getting started with bioinformatics or you already have a lot of experience, we will train accordingly. So for example, if someone is like novice in bioinformatics, so then we have a specific coursework, that is introduction to bioinformatics, that will provide you an insight of what are the different terminologies related to bioinformatics, how bioinformatics can be used in healthcare, how bioinformatics can be used in translational research, etc. Then if someone says that I don't know in which particular research domain or which particular specialization track I need to choose, and so as to complete my research project, for that we have a second coursework, that is bytes and molecules, where we are transiting a participant from a beginner level to an advanced level, where we are describing that participant or a student about the technology as well as the application. So there you will find some of the courses or some of the lessons related to introduction of different omics field, like space omics, like genomics, like transcriptomics, like metagenomics, epigenomics, etc. And so these are the technologies which you learn, a very introductory part. And when it comes to application, you will have lessons related to precision medicine. Then you will have some other components of R language also, then Python language also. So this is how we will train you from a beginner level and then transit to your advanced level, so that you can upscale your knowledge related to your related to the domain in which you have shown interest. Now, here are the topics which we'll cover in the training stage. That is, first, introduction to bioinformatics and the basics of data in life sciences. Second is omics data types like genomics, transcriptomic, or metagenomics, as I told you. Third is data science and machine learning for non-engineers and biologists. And the fourth is R and Python programming and in-depth analytics for complex omics data. So for those who are interested to interpret their results through R or Python programming language, then there would be in-depth analytics for like in-depth review of the courses for them. Those who don't want, there is just an introductory level of courses also, just to introduce you what R and Python is all about. Now, once your training is over, we will be transiting you to the second stage, that is find an impactful research topic. So here you need to learn how to perform literature review and how you need to plan out your research project. That is the methodology which you need to follow so that you can come up with the results and eventually do the interpretation, either statistical or the biological. So there are few points which you need to keep in mind while you are in your second stage and you are performing the literature review. You are finalizing your data set on which you need to work on. You are preparing a flow chart which you need to follow. So what are the few things? First is make your research question compelling to those already in the field. Set some specific parameters to answer the research question conclusively. And third is find data set that fit your objectives and explore them for batch effects or bias. And the fourth is get feedback from our expert mentors on your topic and analysis proposal. So one thing I would like to make you all clear here is that this whole research fellowship program is mentor oriented. It's mentor guided program. So at each and every step, whenever you'll get stuck, you can take help of our expert mentors, and our expert mentors are from either industry or from academia. Not only like mentor, if mentor guidance, you will also have online technical guidance. For example, while going through with the analysis phase, if you find a technical glitch in the T-Bioinfo platform, so we have a particular team assisting you for that also. Now, once your analysis is over, you have results. The last part, results, you have the interpretation, etc. Then the last part is present your analysis for expert feedback. Develop a complete project ready for expert review and feedback. And once your analysis is done, your interpretation is done, you have the novel finding, you need to jot it down everything in a proper manuscript, scientific manuscript or a report. So whatever you have done throughout your research fellowship program, so that you can gain expert review and feedback. Okay, if you are thinking, if you are planning out for any research publication or maybe presenting your research project in any of the symposium or any of the international conferences, so there are some potential outcomes once your research project is done, is completed successfully, and some of the outcomes are listed over here. So the first is expert feedback and overall evaluation of project quality, innovation and analysis execution. Second is poster presentation at the research fellowship meeting or research symposium. And by research symposium, I would like to inform everyone, and rather I would like to invite all that we are organizing a 2 days research symposium in March on 30 and 31st. I'll quickly show you the landing page of research symposium also in the end. And third is path to collaborate with expert mentors or faculty at partner universities. And fourth is path to a full publication, a research publication in a good peer-reviewed journal. Now, there are four different tenures in which one can enroll. So right now we have like on my screen there are three different tenures only which is being displayed, but there are four definitely. One is 45 days, another is 60, then is 90, and then is 180 days. So 3 months and 6 months option is also available. So the resources for all the tenures are common, that is asynchronous courses, hands-on assignments, group sessions, program course access, OmicsLogic portal access, T-Bioinfo server access, then online mentor assistant, online technical assistant, certification after completion, etc. So these are the common resources which you would have access to. The difference is the outcome. Obviously, if someone would enroll for 45 and 60 days, they cannot think of a full-fledged research project which can be publishable also. So the outcome of 45 and 60 days is a literature review, a kind of intermediate level of analysis, and a project report for that intermediate level of analysis. And then for 60 days, it's there is one more addition, that is a research poster presentation. Like they can present their poster in a research symposium. Then for 90 days and 60 days, there is obviously a research poster, project report, literature review, and a research paper publication also. So these are the outcomes, the tenures, etc. Now, when it comes to the payment, I would like to I would like to redirect you to a program page so that I can show that we have different payment options depending upon the locations of our participants. And here I'm pasting the link of the program page in the chat box. So I'll firstly show the payment in USD for all the participants who are there in America or Canada, etc., countries outside India. So uh, so for 45 days it's $350, for 60 it's $450, and for 90 it's $550. And someone who is from any other country, for example from Europe or from Brazil, from India, they can simply click on the currency, and they can see the pricing in their own country currency. So for Indian community, for 45 days it's 9,000, for 60 days it's INR 15,000, and for 90 days it's INR 20,000. And right now I would like to inform you that yes, we are providing scholarship to all those students, like especially to students and also to those participants who are from low income countries. So there is a scholarship opportunity available, and those who are interested, they can either ping me on my email address. Shigori, can you please share the details of me, the email address and the contact number in the chat box, so they can either mail me or they can directly connect me on WhatsApp. So the number which Shigori will share, yeah. So the number which is being shared in the chat box, it's active on WhatsApp, and you can directly ping me on WhatsApp, and we can discuss how much scholarship one could have. So yes, so this was all about the resources, the tenure, the outcomes, the payments, etc. And when you scroll down further, you can check out our students projects also, so on different domains like infectious disease research, then space omics, then precision oncology, then for machine learning and modeling, then related to some skin disease, data-driven drug discovery, etc. And then eventually you can find out our collaborators from all around the world and the mentors of research fellowship program, and then some related blogs about our students' research project, their success stories, their achievements, etc. So this was all about the research fellowship program, which I would like to tell you. And I'll just quickly show you the landing page of research symposium also. Give me a minute. Yes. So I'll again share my screen. Just a moment. Okay, now I guess everyone can see my screen. And Shigori, could you please share the link of the research symposium main landing page in the chat box for everyone to they can go through it.
Yes.
So this is the research symposium page that we are going to conduct on March 30 and 31. It's a 2-day event, and soon we'll share the session schedule also. So for those who just want to be a free listener, they can just simply fill out the form, which is here in the end here here. So they can simply fill out the form, and we'll get the registration, and we'll share the Zoom meeting link accordingly with them. Now, when you'll scroll down, here is what we'll be focusing on, like in the research symposium. We'll be discussing about how one can enable like advances in enabling technologies. Then what are the different career opportunities in bioinformatics or computational biology? We'll be showcasing you research projects through our poster presentation competition, and we'll be having like some exciting cash prizes also for that. And then how one can get involved in finding labs, programs, and mentors. Basically, like getting get like talking to each other, talking to like-minded people, and getting your things reviewed, getting your research project reviewed through expert mentors throughout the globe. So this is the research symposium page. Again, we have categorized the research symposium page into 3 categories basis on the student, then faculty, researcher, and then the poster presentation. So first of all, it is free for the listeners. Those who want a certificate for a 2-day event, so they like it's a very minimal amount of fee that we are charging. That's $5. And if they'll click on this, they'll be redirected to the checkout, like the payment checkout page. They can fill out their payment details. See, so it's just $5 for students, and for students only. So they can fill out their card details and proceed with the payment checkout. For faculty and researchers, it's $10. And for those who want to be a part in a poster presentation competition, so there is a fee of $15. And these are the access which they'll have once they'll pay for the poster presentation, or if they are a faculty or a researcher or a student in under or postgraduate. So this is how you can explore this page. And yeah, so this was all about the research fellowship program, the Omics Research Symposium. And we are like on daily basis, we are rolling out some mails and also posting about the research symposium, etc., on our social media handles. So please do visit the social media handles on either Facebook, Twitter, LinkedIn, or Instagram, and keep yourself updated regarding the same. And also, we have created an event, you can join that also. So, yeah, thank you. If anyone is having any question, please feel free to ping in the chat box, and I'll be there to answer your questions. Thank you, everyone. And Shigori, over to you.
Thank you so much, Sparsh. So we'll pause for a couple of seconds, and if no one has any queries, we'll be ending today's meeting.
So I see one query in the chat box of uh, Darius. I'm so sorry if the pronunciation is wrong. He's asking for, can you resend the link to the introductory courses? Could you please share your email address with us, and we'll be mailing you out about the introductory courses, the links.
All right. I guess no one has any further queries. Everyone is just sharing their email IDs. If no one has any further queries, we'll be ending today's meeting. And once again, you can reach out to us at any time through the email ID and the contact number that we have provided. And with that, thank you everyone for joining today's webinar, and I hope you have a great day. And we look forward to seeing you at the Research Fellowship Group. Thank you, everyone.
Thank you, everyone. So we have noted your email address. We'll be mailing you. Thank you.